on the sides.
So, and then you are also streaming on YouTube if you are interested in that. Okay.
How I know, I didn't know. How am I streaming on YouTube?
No, you don't care.
Ah, okay.
It's done automatically.
Okay, good morning to everyone. It's the third day of the workshop. I will wait a few minutes more before to do the introduction of Luca, our first speaker today. And I think most of the is now getting in.
Leonel, can you hear me?
Perfect, clear. Yeah.
Ah, okay. It is 40 minutes, right? About more or less.
Yes. And it's, I'm watching at your screen, so everything is ready to go. I will introduce you in a minute.
Okay, just let me know if I need to use earplug, but I think it's not an issue. No?
No, I hear you very good and clear, no issues. Perfect. I think we are more or less ready to go. So I will introduce our first speaker, Dr. Luca. He's assistant professor at the Department of Physics and Astronomy, University of Catania, Italy. He was former postdoc at the LFD with Enrico Graton, and also he has a position at the Istituto Italiano di Tecnologia in Genoa. Luca is a world expert in the methods and instrumentation, and in particular within the super resolution and the combination of super resolution with film, another variant of STED and super resolution. Besides that, Luca also have done major work in the image correlation spectroscopy, and he have developed with some of his students very interesting method about this technique. Today, Luca is going to talk about the development of the time result super resolution STED and how the combination of film phasors can help us on this method and this application. So thank you very much, Luca, to be part of this workshop, and we are ready to go.
Thank you, Leonel. thank you for the invitation. It's really an honor for me to participate to this workshop. And unfortunately, we are online. And again, I hope one day to visit you in your place in your lab. Anyway, the topic of today is, as Leonel mentioned, is super resolution and the combination with film phasor, which is, I think, a subject you already discussed a lot in this workshop, as I can see from the different topics we have discussed in the past days. And I will start just with the brief motivation of my current research. And in particular, I'm interested in the process which are involved in the generation of cancer. We know that genomic instability is a hallmark of cancer, but we don't know yet what are exactly the process which after specific mutation, which we call oncogenes, bring some instability in the genome. Probably, according to several theories, there are alterations in the mechanism of DNA maintenance, meaning the replication, the transcription, or the damage response. So I'm currently interested in studying this process. And clearly, at the molecular level, today there is a lot of techniques which are based on genomics, meaning the analysis of specific sequence, which now can be observed also as a function of time. So we know exactly which sequence are replicated or transcribed at different time of the cell cycle. So what we are still missing probably is that this process cannot be studied simply on a linear sequence of DNA. We need to keep in mind that the nucleus of an eukaryotic cell is a very complex organelle, and in which the DNA is long about 2 meters. But these 2 meters of DNA are packed in a very small space of about 10 micron. And in fact, if we look at the genome inside the cell, DNA is wrapped around nucleosome and in a complex machinery, which we call generically chromatin. So, so many of these genomics-based approach disrupt the cell to study the sequence of DNA. It will be interesting to be able to approach the same process by visualizing this process in the intact nuclei. So it's in some ways like an analogy is instead of knowing just a single letter of a book, which will be like the classical genomics approach, we would like also to study how the organization, to understand the organization of an entire library, which involves clearly a more complex three-dimensional organization. So what this has to do with microscopy? Well, because today there are with the development of several labeling strategies, we can today label several points where DNA is being processed. In other words, we can find the point where DNA is replicated or transcribed or is being repaired. And so we typically observe this kind of spot when we label this point and we observe under a microscope. But we know that conventional microscopy is limited by diffraction to about a resolution of 250 nanometer, which is not enough to study many of these processes in the nucleus. So today I'm going to discuss which are the main strategies for optical super resolution, and in particular, what is the approach which is based on phasors. I will start with just mentioning what is the resolution. I think everybody in this audience knows the difference between these two images. I can understand that this image on the right has a clearly higher resolution than the one on the left. But basically, in an optical system, what is that limit the resolution is the optical diffraction. In fact, the common expression is simplified expression is typical of the resolution, so the ability to resolve two objects is given by the wavelength of light divided by 2 times the numerical aperture, which is related to the angle formed by the main optical lens. So in other words, this resolution is the limit of resolution is an effect of diffraction, the diffraction which is imposed by the use of the lens. Another word to state this limit is by saying that even if we have a point source, for instance, a very small molecule which is fluorescent, when we build an image of this point source, because of diffraction, we will not get the point, but we will get an object which is larger, an image which is larger. And we call this image the point spread function, which describes actually the limit of resolution of the microscope. So what we can do? Just to state again, the stress again, this concept, because it's important for what I'm discussing. On the left, there is a simulated image with lower spatial resolution, where we cannot resolve, you can see that we cannot resolve the different point. On the right, we can see an image obtained with higher spatial resolution, where we can resolve the different object. I will say that this system has a larger point spread function, because a point is broad and corresponds to an image of a point corresponds to a larger spot. In this case, we will say that we have a smaller point spread function. So in other words, if you want a better resolution, we want a smaller point spread function from our optical system. But the small PSF, how we can obtain a small PSF? We need to use, typically we use a high numerical aperture objective. Anyway, we cannot get beyond a numerical aperture of 1.5. So the point spread function of a microscope cannot be arbitrarily small because of this limit of diffraction, which was stated by Abbes, and it's called also Abbes law. In practice, the typical value that we obtain in confocal microscopy are in the order of 200 nanometer for lateral resolution and 600 nanometer for resolution along the optical axis. So, how this limit has been surpassed? Basically, we know that the Nobel Prize in Chemistry was awarded in 2014 to these three scientists for the development of super-resolved fluorescent microscopy. And but it is important to stress at this point that they, and of course many other scientists, didn't change the laws of diffraction, which determine the principle, the limits of diffraction, the limit of resolution. So what did they do, and why they get the Nobel Prize in Chemistry? But actually, we can say that they showed, and they and many other scientists show how to play with the photophysical properties of the fluorophore to get a smaller PSF. So they didn't change the laws of diffraction, but just show how play with the photophysics and with the fluorophore to get a smaller point spread function. This will be a scheme to describe the main strategy of super resolution. So this will be a schematic of an object, which can be thinner as we want. If you look at this object with an optical system, microscope must have a lens. The lens is represented here as an objective. There is a limit of resolution represented by this point spread function. The result is an image like this, which is, of course, is poorly resolved because of the size of the point spread function. Now let's come to the main strategy of super resolution. Super resolution uses a process which is fluorescent, which of course you have talked a lot in this workshop. And so we have to keep in mind that the fluorophore are labeling the object, are excited by some excitation wavelength, and then they emit fluorescence. This is important to understand how fluorescent super resolution works. Let's start with the first main approach, which is called single molecule localization. This approach is common to techniques which goes under different names, for instance, storm, palm, or paint, and so on. In this approach, the main point is that we are able to switch only one fluorophore on in the point spread function. And so if only a single fluorophore is on, we can now build an image of a single fluorophore, and we will get an image similar to this one. what is important that if I have only a single fluorophore, I can find the position of this fluorophore in an image with a very high precision. Here you see the image obtained with different number of photons. You can understand from this image that if I have a lot of photons, the precision, the center of this spot is determined with higher precision than in the case I have less photon. In fact, it can be demonstrated that the localization precision is in the order of D, where D decides our resolution, diffraction limit, divided by the square root of the number of photons. So what we have to do is acquire enough photons and localize a single fluorophore, and we can get a nanometer precision. Okay, so this is schematically shown here. I collect an image, I can get to a resolution of 30 nanometer for a single fluorophore. Is this enough to build an image? Well, of course, a single fluorophore will not make us an image. So we need to repeat for all the fluorophore contained in our structure. Because this procedure is repeated in a random way, we cannot control which one is switched at a given time. We call this approach also stochastic switching. And we need to repeat this process in time. So we repeat a second time with another fluorophore, and so on. We repeat several times, several thousands of times. At the end, we are able to construct the signal from all the fluorophore at a higher resolution. So, which is the main limitation of this approach? You can understand that because the molecule has switched and imaged one at the time, we need to acquire many different frames. So in general, this approach is slow because it requires several frames. Here I show you an example. This is an image provided by Simone Pellicci in Milan. This is an image of a nucleus where there is labeled an incorporated nucleotide, which basically labeled the point where transcription is occurring. We call transcription foci. This is diffraction limited. If we now perform this trick of the switching, we basically obtain an image like this. And this is a reconstructed image. To obtain this image with a method called distorm is the result of about 20,000 frames, where each frame is about 30 milliseconds, which is the time required to image the few fluorophore on the image. So this is a very beautiful imaging approach. What are the requirements? The main requirement that we need to switch the fluorophore in a stochastic way. And so there are several approaches that produce this effect of having the molecule switched one at a time. For instance, in STORM, we have activator reporter pairs. In what is called direct STORM, we have standard organic dyes like Alexa 647. If you are interested, you can read this. There are several sources that describe the several strategies that are used to produce a single fluorophore imaging. And so PALM is another use a photoactivator fluorescent protein. PAINT use even another approach. Another issue, which is a common issue in this in STORM or similar approach, is that typically we require the correction for drift. Because the image requires several minutes in general, we need to correct for drift by using some markers to correct for the drift of the sample. Now I will show you what is the other approach, which is based on a totally different strategy. So the first strategy is to separate the molecule in time using the trick of photo switching. Now let's start again with our simple scheme, and I will discuss as a second strategy stimulated emission depletion. In this approach, we have again our point spread function, but this time all the fluorophore are excited. So basically, if this is like what happens in a confocal microscope, we excite the molecule on a given spot, they are all excited, so all the fluorophore are on. How can I get a smaller point spread function with this approach? Well, the general idea of this strategy is to add a second laser beam, which is superimposed with the previous one. And in general, this laser beam has this shape of a donut, so with a hole in the center. The second laser beam has the function of switching off the fluorophore in the periphery of the PSF. So meaning in the center, we have no effect, we leave the fluorophore on. In the periphery, we are able to switch the fluorophore off with this laser. For instance, instead, the effect that produces the switching off of the fluorophore is stimulated emission. That's why there is this name, stimulated emission depletion. In fact, if we tune the second laser to a particular wavelength, which is in some way coincident in a region of the emission spectrum of the fluorophore, we can induce the following transition, indicated here as stimulated emission, as the red arrow. So this transition will switch off the fluorophore, will deplete the signal from the fluorophore in the periphery. Of course, we have to think that if you think of another method to switch off the fluorophore, we are still in the same strategy. In fact, there are other processes which can produce a similar effect, but they do not use stimulated emission. So more in general, we will refer to this strategy as targeted switching. So with respect to the previous strategy, which was stochastic, so the fluorophore were randomly switched one at a time, in this case, the switching is targeted, meaning that the switching is happen in a specific location, happens only in the periphery of the point spread function. That's why it's called targeted switching. Another example of this strategy is Resolve, which uses a different type of process, uses a different molecular transition. So let's come back now to the scheme and what happens if I switch fluorophore off in the PSF. If I'm able to switch them off, you will see that at the end, only the fluorophore in the center will be on. So in practice, this is like to have produced an effective PSF of smaller size. Typically, we reach molecular imaging resolution in the order of 50 nanometer. And so we can build an image which now is super resolved. So we have seen, okay, here is an example of nuclear structure imaged by STED and compared to confocal. So you see that we have seen two different approaches that with two different ways produce a smaller point spread function. An important point of the second strategy, the key point, is that the resolution in this case depends on how many fluorophore we are able to switch off. So in this case, if I'm able to switch off more fluorophore in the periphery, I will get a smaller point spread function and a higher resolution. But what does it mean switching off the fluorophore with the second beam? Actually, it is well known that this is a function of the power of the second beam. In other words, if I use a higher power of the second beam, of the stimulated emission beam, my probability of stimulated emission is higher. So my probability of switching off will be higher and I will increase the resolution. There is also in this case an approximate formula that describes the resolution as a function of the diffraction limited resolution and as a function of the STED, of the power of the STED beam. In this case here there is the square root, I is the intensity of the STED beam, and IS is a constant which is called saturation intensity, which depends on the fluorophore, on the wavelength, and represent the intensity at which we obtain switching off of half of the fluorophore. So you can see from this formula that I need to have a high intensity compared to the saturation value to make the value of resolution of delta smaller. So this is the main difference we compare to the previous strategy. In stochastic switching, we need to collect a high number of photons per single fluorophore. Here we need to use a high power of the second beam. Just to give some number, if I use D as the diffraction limit, 250 nanometer, and here I put an intensity which is about 25 times the saturation value, I get a factor of 5, and I get the number of 50 nanometer, which I gave in the previous slide. So, because simulated emission is a very fast process, the very attractive feature of STED is that it is a process which is extremely fast. So, in contrast with the stochastic switching approach, we have a very fast imaging technique. In fact, it can be used also for techniques like FCS, in which you require a very high temporal resolution. What is the problem? The main problem of STED? The main problem is that it requires the high illumination intensity to switch off the fluorophore. In fact, in some STED, the more common STED configuration, we can reach peak intensity of the second beam in the order of 1 gigawatt per centimeter square. So the problem is that in general, in biological system, having such a high peak value might introduce side effects, such as photobleaching of the fluorophore, phototoxicity, or both. And so in practice, even if the resolution in principle is unlimited, because I can use a very high power, there is a limit that is in general represented by the limit, the tolerance of the sample. So for this reason, there are several strategies to reduce this side effect. For instance, probes, we don't require a special probe that photo switch, as in the case of storm, but we require probes which are which have excellent photostability. Another strategy is to use probe with spectrum in the near infrared to reduce phototoxicity. Other strategies are those which I will show you, for instance, the one that we will see with the phasor, to reduce the illumination intensity, but keeping the same resolution. So now I will go to an oversimplified summary. I guess it's extremely oversimplified for both the strategies. So we have started with this schematic drawing. The PSF of an optical system is limited by diffraction to about 250 nanometer. The first strategy is stochastic switching, one fluorophore at a time. It can be done, it is done, but the main limit is the poor temporal resolution, because we have to switch off, we switch on one at a time, so we need time to image all the fluorophore. In the second approach, it's called targeted switching. An example is STED, and the idea is to switch off all the fluorophore in the periphery. The main limitation, if we refer to STED, is the high illumination power. So this condition, from my point of view, this limitation comes from the fact that we need to switch completely off the fluorophore. Here there is only one fluorophore on, the other are off. Here there are only on the fluorophore in the center and the other off. Now I want to just make you thinking of a different approach, which is the one we are going to discuss. Is there an alternative strategy in which we get the super resolution, but without the need of switching completely off the fluorophore? So this is what I'm trying now to tell you in the following part. Sorry for the animation. Of course, there is always some mistake. So we start again with our diffraction limited PSF, where all the fluorophore are on. Now imagine that we have a different strategy to encode some extra information on the fluorophore. In other words, imagine that the fluorophore in the center of the PSF have some behavior, and the one in the periphery have a different fingerprint. For instance, we can add in the microscope an additional channel. For instance, I can record the time. This time can be the lifetime of the molecule on any other properties, but let's keep the discussion general. And if when I visualize the fluorescence into this channel, let's assume that in the center the fluorophore has a specific fingerprint, while in the periphery they have a different fingerprint. For instance, in the center, the decay of the signal is low, and in the periphery, the decay is faster. When I measure the total intensity from a given pixel, which is here, this black dashed line, now I know that there is a contribution. All the fluorophore are on. I don't need to switch them off. But I know that this fluorophore appear different on an additional channel. How can I get now the super resolved part? I mean, the center of the PSF without switching off. What I need to do is to separate the central component, which here is represented as the fluorophore in green, representing as the fluorophore which have, for instance, this fingerprint in this channel. I need to separate this component from the component of the fluorophore which are here represented in red, meaning the fluorophore which have a different fingerprint in this channel, which are a faster decay. So in this, according to this idea, I don't need to switch the fluorophore off, but I need to separate the fluorophore based on the analysis, on a kind of spectroscopic analysis on an additional channel, for instance, in time. We need to do this analysis for each pixel, and we need to estimate the fraction of photon emitted from the center. This approach is in some way less, less explored than the two-way strategy, and was actually, to the best of my knowledge, first discussed in 2005 by Enderlein, then a few other papers, and then in 2015 we actually introduced our work, which explain how this can be useful in microscopy. So now, at this point, I just remind you what is the phasor approach, but I guess that you have seen a lot yesterday. So the phasor approach for us is just a way to analyze this kind of spectra. So if I have a decay which is lower and this one is faster, I will get a different phasor, which is basically obtained through the calculation with the Fourier transform. So it's the frequency representation of a time domain spectra. Okay, here is how we can remember the position. Imagine we have a speedometer like the one of the car, but we have a faster decay on this side of the phasor, a slower decay on this side of the phasor. Now I will go back to my scheme. In this case, what we want to do is to extract the fraction of fluorophore, the fraction of the signal corresponding to the center of the PSF, meaning the fluorophore which have the specific fingerprint, using the phasor. I am sure you have seen in these days decomposition in the phasor approach. We can use decomposition to calculate fret, fraction of donor in fret. You can decompose spectral emission. So in general, the importance of phasor in microscopy is that it's a linear approach, and every time we have a signal represented here by generic spectra, the intensity here, and I have the corresponding phasor here in black, if I know that this phasor is the spectrum, there are two components, I can calculate, I can decompose this phasor linearly into a component which I indicate here P center, and the component which I indicate here phasor of the periphery. It is well known that in the simplest case, the fraction corresponding to the center is related to the distance of this point along the line that connects the phasor. So in other words, in this alternative strategy, we will use the phasor approach to extract the fraction corresponding to the center of the PSF. So this is the connection between phasor and super resolution. We need a strategy to encode information inside the PSF. And then we will use the phasor to decode this extra information using this simple decomposition in the phasor plot. How we build the final image? Well, the final image, which I'm calling here split, just to remind you that I'm splitting the PSF into parts. The final image, which is the one which is super resolved, will be calculated as the fraction which I calculate in the phasor approach multiplied by the total intensity. So in this way, I will calculate only the photons which correspond to the center. Why split? So this name, split, I saw that there is also a movie which refers to split of personalities. There is also a city in Croatia. In our case, split, we gave this name because the first implementation, which is the main one I'm discussing, was based on variation of fluorescence lifetime. In other words, we have used a steady beam to induce a difference in lifetime within the PSF. In other words, in the outer part, fluorophore have a shorter lifetime. In the inner part, they have a longer lifetime. That's why we call separation of photon by lifetime tuning. And this was an article which we published together with Enrico, with Alberto Diaspora, Giuseppe Vicidomini in 2015. Here I explain you this method more in detail. We need pulsed excitation to observe the fluorescence lifetime. I'm sure you're talking about FLIM. The fluorophore inside the PSF normally have all the same lifetime, but we need a strategy to modify the lifetime. This strategy is provided by the addition of a steady beam in continuous wave. Because stimulated emission is an additional process, an additional pathway for decay from the singlet excited state, the depletion laser modifies the fluorescence lifetime. I'm not writing here the formula, but it's important to know that the lifetime is changing depending on the power of the stimulated emission beam. So the higher the power, the higher the shortening of the lifetime. So this is the effect. When I add the steady beam, I have a lifetime which is the same in the center, is long, is not perturbed, but in the periphery, I modify the lifetime. And so I get something like this. If I observe the nanosecond, I will get a different fingerprint in the time domain, or equivalently in the phasor plot. How much do we need to modify the fluorescence lifetime? Well, we basically, the power that we need to use with respect to pulsed studies in the order, it's about at least one order of magnitude lower. And this is the same order of magnitude that we use in a technique called gated STED. You will see here that from an image of beads, you can observe the effect of your STED beam that is causing here a spreading on the phasor plot corresponding to this effect of center and periphery variation of lifetime. In principle, we can use also a very small, a very lower power of STED, but we need to keep in mind if we use a very low power, the variation will be smaller. Can we work at this low power? You will see here that the phasor is more round. I will say that we can also work at this low power, but we are limited by the signature ratio. So if we want to perform a measurement at very low power, we need to acquire more photon to be able to resolve these spectra, which now are much similar. Here now I'm showing an example, again with transcription foci. This is a confocal image. And here I'm showing after I turn on the STED beam, a continuous wave STED beam, you will see here that if I build a lifetime image, I observe lifetime variation. It is the same fluorophore in the same environment. Here the variation are due to the action to the process of stimulated emission in the different pixel. When we process this image to extract with the phasor plot the fraction of the center, so I can build this final image, which you will see is super resolved, and it is obtained by multiplication of the total signal by this fraction of the center, which is calculated by phasor. So here, up to now, I'm showing you that with the phasor, we can extract, we can obtain a super resolution by exploiting information into this additional channel, which is the lifetime of the fluorophore, modified by the steady beam. There is an important point of phasor which I did not discuss, which is that when we do a decomposition, we can do it several times. We can do it in several ways. So basically, in this scheme that is shown here, the phasor of the signal is decomposed into two components, center and periphery, but there is also a component which corresponds to the origin of the phasor plot. And this, we typically call the uncorrelated background, because it's a phasor of this position is something which is flat in time. And so when we do this decomposition with 3 components, we obtain center, periphery, but also this fraction of background. Why this is important? So in the example that of the image of the nuclear foci, we get 3 images typically. We get a fraction in the center. This one is the image that which is more interesting, is the super resolved image. But we also get a second component, which you can see here from the screen, has a shape typically of rings. And then we get also this component of the background. Now the point is, what is this background? Why we get the background? Is the background from the room or is something else? Well, instead microscopy, this background can be an important unwanted effect. In fact, instead, one of the side effects is that the STED beam, which has such a high power, can be absorbed. So instead of producing stimulated emission, produce unwanted effect. If the STED beam is absorbed, it can produce an unwanted signal, which degrades the quality of the image. What is important in the context of pulsed excitation and continuous wave steady is that any effect arising from the absorption of the STED beam is continuous in time. So this effect, this noise, this background is constant in time, has a fingerprint which is constant in time. Here I show you an example where you see a sample where this background effect is in some way not negligible. Here there is a confocal image. Here is the phasor of the confocal image. When I turn on the STED beam, this is the lifetime information. You will see that there is information which is useful, but if you look carefully at the phasor plot, you will see that the phasor is moving toward this position here, which I told you is the position of the background, the uncorrelated background. This effect is due to an unwanted effect of excitation by the STED beam. This can be seen also if we plot the average decay. You will see that we can estimate how much is this effect of the background. What is the effect on the image? Well, when I perform the split approach, which means the calculation by phasor, we will see that the split image doesn't contain any background. So this is good. The split has not the background. Why? I will compare here with the standard technique, which is gated STED. In gated STED, we can acquire the data in a similar way in a time-resolved STED microscope, but you will see that the gated STED image contains a lot of background. Why? Because the simple gating operation consists in acquiring the signal after a given time delay that we call time gate. But because the background is this flat contribution in the bottom, when I perform gating, I keep the background. So the final gated STED image is degraded by the STED direct excitation. Our approach, the split based on phasor, does not contain background because it's based on a separation of component. This one is the slow component of the signal. The second is the fast component. The background is the flat component, which is due to this unwanted effect of STED. So this is showing an advantage of using the full decomposition by phasor compared to a simple gating approach. So this is the main conclusion, I would say, of this part, which is that using the phasor, we can get an improvement of spatial resolution. We are using the information encoded in the lifetime. But we can also get a second advantage, which is the removal of background due to the direct excitation from the STED beam, because we are performing a true separation of components. And in fact, the similar strategy is currently now employed in what's called the tau STED, and is employed in the new microscope commercialized by Leica, which I guess is substituting in some way the gating approach, is improving the gating approach, introducing this phasor-based decomposition. Okay, for the sake of time, I cannot describe the other development, but just to mention that this approach based on phasor is not limited to what I described here. In the context of STED, we demonstrated at least the other two approaches. So we can perform the phasor, the super resolution, in the concept of pulsed STED. So in this case, we have pulsed excitation, pulsed STED, and we need still the lifetime detection. And another approach is based on the modulation of the STED power. So basically, we can just obtain a similar effect without the lifetime by considering an additional channel. With this, I conclude. In summary, I show you what are the main strategies for super resolution based on switching off, stochastic or targeted. And then I showed an alternative strategy based not on the switching off, but on the separation of fluorophore within the point spread function based on a difference encoded into an additional channel. And the phasor is a very practical tool to perform this decomposition. Instead, microscopy, the main advantage we demonstrated is the improvement of resolution using a lower STED power and the removal of background. With this, I just acknowledge the collaborator at the Italian Institute of Technology, with I performed most of this work. Also Enrico Sirvain, collaborator at the European Institute of Oncology, and now I currently moved to Catania, to the University of Catania Department of Physics, where I'm setting up my own lab, and I'm ready for questions. Thank you.
Thank you very much, Luca.
Luca. Yes, Luca, I have a question. So there are processes which depends on distance, of course, FET, but there is also metal-induced decay or metal-induced fluorescence. Do you know if anyone used the metal-induced fluorescence in order to do the same process and separate the fluorophore which are close to the metal from those which are far from the metal just directly by lifetime?
So there is this is a technique called, I think, MIT by Jörg Enderlein,
yeah,
where basically they use the influence of the metal
but talking about, I don't know if you remember when you were in my lab, Laura Estrada was using gold nanoparticles in the nucleus, and then
yes.
We measured the lifetime many times, and that was, the lifetime changes a lot from one point to another, but never that, she never developed in an imaging method. The surface, I understand that part, but for example, you can have something which can go in the nucleus, while a surface cannot go in the nucleus.
No.
So do you know of anybody that continued this kind of work?
Enrico,
okay.
Yes. There is a group, I don't remember now the name, who is using some surfaces with sparse metals, and they can measure the distance based on the.
I know that, Lionel, but I said in the nucleus, for example, you cannot put those things in the nucleus. So what's intended to be in the nucleus? Something that can roll on the nucleus, or can roll on the chromatin and map the entire chromatin. It's a different idea.
Yeah, I understand what you are meaning, right? Trying to find, because the range of effect that you have is very close. So the
distance determines the lifetime, and so things which are very close will be more quenched than things which are very far. But that is very different than the effect of light in the beam. So you don't need the donut, you don't need anything. You just measure the phasor essentially.
You just need you need this gold nanoparticle to move randomly.
Yeah, but we know how to move that, and this is the work we did with Laura. We can move the nanoparticle everywhere we want.
We can shoot the nanoparticles of gold.
Well, we can move also due to thermo effects.
Yeah, remember.
Yeah. So anyway, I want to know if anybody has continued this work, because since you left the lab where we were measuring the lifetime, essentially, that was another application of the lifetime and was never done. Okay, so maybe I can convince somebody to do it.
I am looking at the chat. There is no question. Let me see if in the participant there is any hand to make a question. No, we don't. Here. There is a question from Sebastian Acuña. What is the reported resolution for the split?
It depends on the, of course, on the state power that you use. So there are, I would say, there are not so many paper on this. But based on my experience, we get the order of 70 nanometer with the lower state power. So it's in some way an advantage of having a lower state power, but still having a good resolution.
Okay. Great, Luca. Thank you very much for the talk and participating in the workshop. If you can stay later, we will assign the people who has more questions to you in a break room. And if not, I can pass your information and the people can contact you in any time.
Okay.
So move on to the next talk. Fernando is already connected. I think you can share your screen, Fernando, and unmute. Meanwhile, I will introduce Fernando. Dr. Fernando Stefani is professor at the Department of Physics in the School of Natural and Exact Science in la Universidad de Buenos Aires, Argentina. He is a principal investigator in the Consejo Nacional de Investigaciones Científicas y Técnicas, CONICET, in Argentina, and he is vice director of the Center of the Biophotonics Research. Fernando has received numerous awards and recognitions across the Argentina and in the world. He has found his lab after came back for more than a decade in Europe, working in different centers, Barcelona and Germany. And he has the chance to collaborate very close with many important people around the world. One of those is Stephen Held, Nobel Prize, as Luca Lanzano mentioned today in the talk. And with this, he began a strong collaboration in the development of a new kind of super resolve microscope, which is called the MinFlux, that can reach few nanometers of resolution. And I think Fernando will explain better about all these possibilities. I would like to thank Fernando and pass you the word for the presentation.
Hi, can you hear me well?
Very well, Fernando.
Okay, well, first of all, thank you, Lionel, for organizing this meeting and giving me the chance to tell you about our latest works. And I also want to apologize for not taking part more on this workshop. I mean, in Argentina at the moment, we are still in a kind of a lockdown due to this, the COVID-19 pandemics, and there are no schools, and I have 4 small children at home. So at the moment I have to take care of the kids most of the time. We are splitting times with my wife, and actually during the mornings I have to take care of the kids. So now I have negotiated some change of shift to be able to give this talk, and I hope to be able to stay for the rest of the meeting, at least on my cell phone, to listen to the rest of the talk.
I appreciate it very much.
Disclaimer done. Let's start with the talk. So I think the talk by Luca just before me will save me a lot of time. So I actually, while I was listening to him, I just rearranged quickly some slides to pass quicker all the explanations about fluorescence nanoscopy and maybe move faster to some new methods. Okay. So as Leonardo said, I work at the Center for Bio-Nanoscience Research. It's a new institute in Buenos Aires. It was created in 2012. It's a small institute. We are 10 PIs working on very different subjects, all related, of course, to bio-nanosciences, a lot of chemistry, nanochemistry, supramolecular chemistry. And well, my group is the one working on nanophysics. And we are a small group. So this is a snapshot of 2015, a more recent snapshot of 2018. So we are a small group of postdocs and students, but we have a powerful network of collaborators, both nationally and internationally. Our current research is basically split in these 4 lines, like optical manipulation, we call optical printing of nanoparticles, self-assembled nanophotonic devices using DNA origami technology, nanoscale temperature measurements, and fluorescence nanoscopy. This is the subject of today, and in particular, I will be talking about new methods of nanoscopy. So the work I will present today has been done in collaboration with Stefan Hell, with Damián Refojo from Buenos Aires, with Alfredo Cáceres, Nicolás Unzain, and Mariana Bisbal from Córdoba in Argentina, and with Guillermo Acuña from the University of Freiburg in Switzerland. Okay, so all this, we are going to skip it. How good is fluorescence nanoscopy? We all know. We know what's good for, what the limitations are. We know about the diffraction limit. So I will just want to stress the size mismatch that we have between our photon sources, between our fluorophores, our molecules, 1 nanometer in size, roughly, and the image of such a fluorophore in a microscope is about 200 nanometers. So we have a size mismatch of 1 in 200. It's a very large size mismatch, so there's a lot of information that we miss if we do conventional imaging. Okay, so let's move on. So fluorescence nanoscopy was invented. A first generation of methods provides a resolution in a few tens of nanometers. There are 2 families of methods. Luca just mentioned the coordinate targeted nanoscopy and the coordinate stochastic nanoscopy. So how does it work? We are going to skip it. I will do a break. I will stop here just to explain DNA origami, because this is a technology that we use a lot also to test new nanoscopy methods. So how does it work? A DNA origami is basically a way to construct nanoscopic objects by molecular self-assembly using the characteristics, the properties of DNA. So DNA, you know, forms double helix in a specific way depending on the sequence. So DNA origami exploits this property in this way. So we start with a single-stranded DNA that is schematically shown here by this circular DNA. It's single-stranded, usually taken from a virus or something like that. So it's something like 6,000, 8,000 bases, single-stranded DNA. We, of course, know all the sequence of it. And now imagine that we can mix it up with a small oligonucleotide, just maybe 40, 50 bases long, that half of it is complementary to this region, like the green with the green, and the other half of the oligonucleotide is complementary to another region of the long DNA strand, like this one. So red is complementary with red, green complementary with red. If we let these 2 molecules hybridize, then they will form this structure. So the large DNA molecule will fold in this way, trying to accommodate as many complementary bases as possible. Now, if we do this not just with 1 oligonucleotide, but with many, something like 200, 300 oligonucleotides, all of them very precisely designed to fold the original DNA, the scaffold DNA, in a particular geometry, then we can do that. And then automatically, by self-assembly, we can form nanostructures with a well-defined geometry. And in addition, we know exactly on this structure the sequence of DNA on each position. And this is how it works. This is, again, a rectangle. These are AFM images of these zillions of rectangles that are formed by a self-assembly. But in addition, as I told you, we know the sequence of each position of this DNA origami. So we can let these short oligonucleotides that we use to fold the large DNA molecule, we call them staples, right? So we can let some of the staples longer. For example, this one, we can leave it here. Can you see my cursor? No, I can make a laser pointer. Okay, so for example, this staple, we can leave it at this position a little bit longer with an extra DNA sequence that we can use afterwards to bind at that particular position a fluorescent molecule, for example, or a nanoparticle. So the DNA origami, it can not only form these funny nanostructures, but it can be used, we use them as nano breadboards, like nano optical breadboards to put here light sources, nano antennas, or different components to produce different optical effects. For nanoscopy, we will place basically fluorescent molecules in an organized arrangement. Okay, let's move on. So here you see an example of that. So we have, this is a DNA origami that is has a cylindrical shape, and we can leave these black DNA chains are like single-stranded DNA that we leave protruding out of the nanostructure on specific regions. And we do it like to provide spots for DNA paint. So these are spots where other fluorophores or other oligonucleotides, now fluorescently labeled, can transiently bind to these short DNA chains. And in this way, they can, they come here, they bind for a while, we can make an image, they go, then they bite somewhere else, they go. And in this way, we obtain the blinking necessary to do a super resolution image. And for example, this structure can be used as a calibration sample for super resolution microscopy. Okay, this is an example of this structure. You get the blinking that you need to do stochastic nanoscopy. And if you analyze this, then you get this video is converted into this image where you get all these. Each one of these bright spots is in fact 3 spots organized in a line and with the separation distance according to the DNA origami design. Okay, so targeted nanoscopy has been already explained. So let's move on. So here, let's make just one conceptual note. So in this kind of nanoscopy, like wide field, right? We detect each emission spot and we extract all the information from the emission. So all the illumination is uniform. So we are not injecting any information into the problem with the illumination. We illuminate with a uniform intensity. There's no information there. We collect all the information from the emission of the molecules. We get these emission patterns, we can fit them, for example, with a Gaussian function, and can determine the center. To do this properly, we need a lot of photons to have a good fit and to have a good localization. That's the limitation of stochastic nanoscopy. We need a few thousand photons to get into a 10 nanometer resolution regime. In coordinate targeted nanoscopy like STED, the situation is kind of the opposite. Why? Because we are injecting all the information with the illumination. No? We have this kind of illumination pattern where we have a excitation focus and the depletion focus, and we are going to move these 2 laser foci over the sample with a piezoelectric scanner, for example. And then we can stand in one position, wait a certain time, and ideally, if I collected 1 photon, I could say with some statistical certainty that at that position there is a fluorescent molecule. And then I can move this pattern to another position, wait a certain time, and if I collect a photon, I can say, okay, there is a molecule here, or not, with some statistical confidence. That would be the case if there would be no background, for example, because each photon I detected, I can assign it to a fluorescent molecule. But in real life, there's always background. And in real life, also this depletion laser is never perfectly efficient. So at the end of the day, again, we need a strong fluorescent signal to claim that there is a molecule at that position, because it has to emit above the background. And so both techniques end up being limited by the photostability of the fluorophores. That's, I mean, these techniques, they don't have a theoretical limit. They have a practical limit due to the limited budget of photons that we have from each single molecule. So for that reason, it makes sense to try to come up with some idea to detect molecules in a more efficient way. So this first generation of methods can deliver lateral resolution that is between 15 and 60 nanometers, and an axial resolution that is about 3 to 4 times worse, in the 30 to 120 nanometer resolution. So nevertheless, this is wonderful. You can see a lot more than before. Like here, you see these microtubule bunches, and that when in a diffraction limited image, you cannot discern very well where is one microtubule from the other. When you do nanoscopy, you can now see very nicely the pathway of each microtubule. But again, as in confocal microscopy or normal microscopy, if we zoom in one of these microtubules, we get this kind of images. They get blurred again because we are at the limit of the resolution of these methods. And so we would like to see better than this, because we know that microtubules are actually a supramolecular protein structure, a quasi-crystalline structure of tubulin, of alpha and beta tubulin, forming this quasi-helical structure. So there is organization there, and we cannot see it. We cannot see it. We again don't have the necessary resolution to see the molecular organization. And we would like to do that. And to do that, we need another push into the resolution. We need to get really into the sub 10 nanometer regime, and then we would get really in a resolution regime that is comparable to the size of the structural proteins, and probably we can start seeing these kind of structures in the cells. So that's what we want to do. Okay, so this I told you. Let's move on. So one way to do this is a method called MinFlux. MinFlux is you will see it's actually a very simple method conceptually. Instrumentally, it can become complicated, but conceptually, it's a kind of triangulation problem. So what we are doing is illuminating, we are combining the injection of information with the illumination with single molecule localization. So we are illuminating the sample with some excitation field that is position dependent. Let's see it first in one dimension. We want to locate this molecule, this green star. We want to locate it on this dimension X. So we illuminate this with a field that depends on position. And then when the molecule is here and we use this excitation field, we will record a certain number of photons, N1. Now we switch off this field and we switch it on, displaced along the direction we want to locate the molecule. Now the molecule is excited with a different intensity, in this case higher, so we will record a larger number of photons, N2. So you will see that now N1 and N2 are encoding the position of the molecule. So we can actually establish an estimator that will give us, based on N1 and N2, will give us with maximum likelihood, for example, the position X of the molecule. That's the way MinFlux works. The only particular thing about MinFlux is that we discovered that using zeros of intensity, using excitation fields with zeros of intensity instead of maxima or other shapes, using zeros of intensity, you become very, very photon efficient. So you can detect the position of the molecule with a minimum number of photons. Okay, so you can establish now the statistics of this is, of course, a binomial probability distribution. And then you see that here in light blue, you see the P, the parameter of the binomial distribution that has this shape. So of course, this method is limited in some sense. It works in a one-to-one fashion only between the position of the two zeros. So if I know the molecule is between here and here, then P goes from 0 to 1. The problem is well defined, and I can determine the position. If I cannot assure that, and the molecule can be outside this region, then the problem is not well defined because I have two positions that could have the same likelihood estimator. So in some way, MinFlux is sacrificing a field of view to provide a very high efficiency in the number of photons to locate the molecule. So okay, you can extend this to multiple dimensions, of course. And in practice, for example, to do it in 2 dimensions, it turns out that you need 4 zeros of intensity to do this the most efficiently. And how do you implement this on a microscope? Well, the first way this was done was like this. So in principle, this looks like a confocal microscope, but with some modifications. First, you need this VP. This is a vortex plate to introduce a phase modulation in the beam in order to produce this focus with a 0 of intensity at the center, like instead, the same way. But now we are using it for excitation, not for depletion. We are exciting the sample with this donut-shaped focus. And the other thing we have to do is to shift this focus between these 4 positions. And in this case, this was done with an electro-optic scanner. So basically, first you illuminate the sample with a 0 at this position, you record some number of photons, you switch it off, you switch it on a different position, here for example, you collect a second number of photons, you switch it off, switch it on here, again you record a different number of photons, you switch it off, you switch it on here, and you get the 4th number of photons for this molecule. And now with these 4 numbers, N1 to N4, you can estimate the position of this molecule. That's it. The complications of this setup is that you have to do this very fast, right? Because if you want to make this compatible with the blinking, then you need to shift these positions very fast. That's why we used an electro-optical scanner in this case. So I told you this is very efficient. Here is the proof. These are the simulations, the Kramer-Rao lower bound to locate a molecule inside this area using just 500 photons. And you see that inside the mean flux field of view, so to say, the precision is between 1 and 3 nanometers. Very, very good precision with 500 photons only. And of course, we can do this experiment by moving a molecule with a piezo scanner, for example, and compare the set position with the measured position. And this is the result on the same color scale. So indeed, it is very efficient. You can locate the molecule with just 1 or 2 nanometer precision inside the field of view. The localization error is not isotropic due to the geometry of the field. Okay, but these are technical details I can discuss if somebody's interested. And of course, it's also position dependent. So it's very, very good near the center, and it becomes worse as you reach the borders of the excitation field. But in all cases, it's very, very precise. Here you have, for comparison, the Kramer-Rao bound for a camera-based single molecule localization with a signal to background ratio of 50, which is actually very, very hard to reach in a real experiment. And this is like 2 times better with the same number of photons. Another way to show this is in this kind of plots, like the localization precision as a function of the total number of photons. And we still have the typical inverse law with the square root of the number of photons, like in a camera base, but mean flux provides an additional knob to the experiment. So we can, for example, if we use a field of view of 150 nanometers, we get this curve, way better, like we can get like a 3 nanometer precision with 1,000 photons. But if that's not enough, we can reduce the field of view. And with the same number of photons, we are much better. So mean flux is a tool that is adaptive and is highly precise to look in regions like smaller than the focus. So if we really want to look inside the focus, you can use a mean flux to do that. And it's a technique to locate molecules. So you can use it either for nanoscopy, if you combine it with blinking. For example, inside here, if you manage to make the molecules blink, then you can locate each one of them one at a time and then construct a nanoscopic image. Or you can use it for tracking inside this area to make a nanoscopic tracking, or you can do it for tracking over larger scales if you periodically recenter the excitation pattern. So for tracking, it also has advantages because since we need fewer photons to locate the molecule, then we can track faster, obviously. So here you have a performance example using a DNA origami. So For this demonstration, we constructed a DNA origami with this geometry, where we placed 8 molecules separated by distances of 10 to 12 nanometers, a very small origami. And this is the data you collect. So MinFlux delivers positions. So you can plot it like this, or you can plot it in this way, like in a false intensity map to make it look more familiar to a fluorescence image. And this is an even smaller origami with distances between 3 and 6 nanometers. And you can clearly resolve those molecules. Okay, and this is an example for tracking. Inside a bacteria, in this case, we were tracking this protein, the 30S ribosomal protein, fused to a photoactivatable protein, the MEOS2. And in this experiment, we chose to use just 9 photons to locate the molecule. So every 9 photons, we get an estimation of the protein position with a precision of 48 nanometers. So this is very decent precision. So it's not 1 nanometer, but we are using only 9 photons. And that allows us to go very fast. So we get a position every 125 microseconds using fluorescence, using the count rate of a fluorophore, of a fluorescent protein, actually. And that allows us to do this kind of experiments like you see here. We get a diffusion coefficient with a sliding average window of 35 milliseconds. So this is like very comparable to the frame rate of a video camera. But instead of having 1 position, we get like 80 positions. And that allows us to get a diffusion coefficient at video rate. And then we can say, okay, we can see how the molecule is diffusing with a certain speed. Then it gets stuck in 1 position. It remains there for some time. And then it starts diffusing again much faster. And then it gets stuck in a different position and so on. Okay, so that's MinFlux. That was the first implementation of MinFlux. It has since then been extended to 3 dimensions with the same concept by the group of Stefan Hell. And recently, we just finished building a new implementation of MinFlux that is experimentally simpler, and it also provides access to lifetime information. And we called it the Pulsed Interleaved MinFlux, or PMinFlux. And it's very simple. You will understand it very quickly. So basically, we are doing the same, but instead of scanning this donut-shaped foci with a scanner, with a fast electro-optical scanner, what we do is the following. We just take a pulse laser, we split it in 4, we introduce 4 different time delays between the 4 pulses, and then we recombine them and send them all through the phase modulation vortex faceplate, but we focused each one of these beams on different positions of the sample. And now the excitation pattern of MinFlux is completed in 1 repetition rate of the laser. So this goes at the maximum possible speed. And we detect each photon with time-correlated single photon counting. So now we know from each detection photon, we can assign each detected photon to which excitation donut, with which excitation donut it was produced. And then we get the information for localization. And at the same time, we get the information for lifetime. To do this, we also developed an open source Python code to control a MinFlux setup and to analyze MinFlux data, both conventional and time-correlated single photon counting data for MinFlux, and also to simulate a full MinFlux experiment. So, and this is will also be available with the publication. It's actually already available. We are just finishing commenting and making it more friendly to other users. Well, this is demonstration of performance. Of course, it works just as well as MinFlux. So no, there are no surprises there. You see, here we are locating a gold nanoparticle with 50 photons. We get the accuracy of just 10 nanometers. And if we use a few thousand photons, then we get into the 1 nanometer regime as expected. This is single fluorescent molecules. Again, this is a confocal image. If we zoom in there, it looks like that. Now, if we perform min-flux there, then it looks like that. I don't know if you can see it, but over there you have a few localizations of min-flux with just 1 nanometer resolution. Okay. This is another demonstration of P min-flux to make imaging. So again, in this case we use again a DNA origami with 3 molecules, and we can locate them with this precision. You see the scale bar here is 5 nanometers, and this technique gives you automatically film, right? It gives you lifetime images. So this is a technique that can really give you lifetime images with molecular scale resolution and with no distortions, because this technique does not distort the lifetime of the molecules like instead. So you really can access the clean lifetime information. And you can use it. This is another example where we tested the pulse min-flux for tracking and multiplexing based on lifetime. So these are 2 DNA origami structures that are designed to bind transiently to 2 positions. So this is a long DNA strand with a fluorophore that can bind to this position and to this position transiently. This one, for example, has 8 complementary nucleotides, and that gives us a time of 2 seconds, more or less, 2-second residence time on each position. And this is a faster structure with only 7 complementary nucleotides. So every time it binds, it remains there for half a second, more or less. And okay, we can track these positions in time, like you see here. No, we detect, of course, the 2 positions, but also we use 2 different fluorophores in this case, ATO 542 and Psi3b. So we can discern these 2 structures, for example, based on the fluorescence lifetime or on the average binding time, like you see in these 2 very clear, distinct populations. This was just an example to show the possibility of multiplexing based on lifetime for min-flux. Okay, now I'm going to show you very quickly 2 other techniques that will give you information in the sub-10 nanometer region. This one is called simpler for supercritical illumination microscopy photometric set localization with enhanced resolution. Very complicated name, but what it allows you to do is for stochastic nanoscopy like storm, palm, or DNA paint, you get this kind of images, right, of microtubules. Well, with this simple analysis tool and simple measurement tool, you can get cross sections like this. We can now do cross sections of these microtubules, very clean cross sections, because it provides set resolution that is comparable to the lateral resolution. For storm, we get something like 15, 20 nanometer resolution, and for DNA paint, below 10 nanometer resolution in set. How does it work? It basically consists of using a TIRF microscope in the best possible way, in a quantitative way. So probably you all know TIRF microscopy. What you do is you illuminate your sample with light coming at an angle larger than the total internal reflection, and therefore the excitation light does not penetrate into the sample. You have an evanescent field that decays exponentially like this. Fine, this is well known, and it actually has been used to obtain actual information based on this phenomenon. What we did was to combine this with an additional effect that is also set position dependent, and that is the emission of the molecules. So the emission of molecules close to an interface is also modulated by the presence of the interface. Here, what you see here is the emission angular pattern of a single molecule oriented perpendicular to the water glass interface as a function of the distance to the interface. When it's 250 nanometers away, it looks like that. And as you get closer, more and more light is emitted into the glass emispace, and therefore into your objective. The same happens for a molecule that is oriented parallel to the molecule, with a different angular pattern, of course, but more and more light is emitted into your objective. This can be calculated. This is the curve for a perpendicular dipole. The blue one is the curve for a parallel dipole, and the red one is the isotropic average for a molecule that is rotating during our integration time. So you see that in the end we have two effects that make molecules closer to the interface brighter than molecules farther from the interface. So what we did was basically to compute these two effects carefully, and it looks like this. So we can normalize this to the emission of a molecule at the interface. That's the N0 is the number of photons of a molecule placed exactly at the interface. So the number of photons of a molecule at any axial position normalized to a molecule at the interface has this follows this function. In red you see the exact solution, and in blue you have an exponential fit to it. So it's almost exponential. And for practical reasons, you can use just the exponential approximation, which has this shape. So basically, if you know these parameters, df and alpha f, and you measure the emission of a molecule at the interface, this can be measured, N0, simply by throwing, for example, labeled antibodies on the cover slip and measure their intensity, like here. This is the histogram of emission intensity of molecules just spread on a glass cover slip. So just with these three parameters, you can compute from one measurement of intensity, you can compute the axial position of molecules. And this is what is done, because in stochastic nanoscopy, what you do is you record images of molecules with a camera, and then you have single molecule emission events like this one. A molecule switches on, emits for some frames, and then it switches off. Well, the only care that we are taking is that we are discarding the first and the last frames of this event, because during those first and last frames, we cannot be sure that the molecule emitted during the complete integration time. So we discard these two, and from the rest of the frames, we compute an average number of photons. And with that number of photons and the calibration that I just showed you, we directly assign an axial coordinate to that molecule. The lateral coordinate is obtained, of course, from the Gaussian fit. So with the same kind of data and with a simple calibration, you can get a 3D image of this quality that is actually only rivaled by 3D mean flux or very sophisticated 4-pi setups. Okay, so if we compute the resolution, we have a better resolution we have sub 10 nanometer resolution in the 3 dimensions. And I showed you that one would like to, for example, see the structure of microtubules, right? Now we have the resolution to do it. And this is a cross-section of a microtubule. If we try to identify this kind of helical structure, it's not possible. It's not there. But it's not a problem of resolution. It's a problem that the labeling is not dense enough. So placing, if I really want to resolve this, I need to place one fluorescent molecule every 8 nanometers. And that's not simple. I mean, due to electrostatic or steric hindrance, this is usually impossible. But nevertheless, we can compute statistics, for example, of distances to the first 5 neighbors in the XZ plane or in the XY plane. And if we do that, we see histograms peaking with a separation of about 8 nanometers in both cases, showing that even though the labeling is not perfect, the resolution is enough, and we can even indirectly tell that there is underneath there is a structure as expected. Or in cases that we don't know, we can even start inferring whether there is a structure or not at the molecular scale. Okay, what's the time? I think I still have a few minutes. So I'm going.
You still have, I will say, around 5 minutes.
Okay, perfect. So these slides, I just pasted them during Luca's talk. So they are not very well organized, but I wanted to share with you our latest experiments on STED-FRET. So you all know STED by now, and you all know, of course, FRET. And it's interesting. It would be nice to combine them because FRET provides, again, sub 10 nanometer information, and STED provides information in the, let's say, 20 to 60 nanometer range. So when one does FRET imaging, one is obtaining information about molecular interactions, but it's impossible to say where those interactions are, because the information is an average over a diffraction limited spot of 200, 300 nanometers. And so we obtain average information, which is in many cases is useful and very nice, because it's good to have an average information. But if one wants to know where these molecular interactions are placed and what's the heterogeneity of it, that's not possible. And that's the reason why this is something that has been identified from the beginning of the super resolution microscopy. I mean, the advantages of combining FRET and super resolution, there are papers written about it, but so far there is no, to our knowledge, no method of, let's say, general applicability to combine these two techniques. And we believe that this can solve this problem. So basically what we did was to combine STED and FRET using an intensity-based measurement. And I will show you. The setup is basically a two-color STED setup. There's nothing new here. So it's compatible with any two-color STED. The first thing we did was, we used DNA origami again to test that this was working. We used an origami like this one. It's a cylindrical structure with two spots separated by 180 nanometers, two FRET spots. Each FRET spot has two donors and one acceptor. You may wonder why two donors and one acceptor, not just a single pair. Well, the reason was simple, because this donor was bleaching too fast. So we wanted to be as close as possible to a single pair FRET, but also be able to collect statistics in a reasonable rate. So we just put two donors. In this case, the donor was ATO 594, and the acceptor was ATO 647N. Two dyes that are suitable for STED. And also they have a reasonable overlap to do FRET. So what you see here is the two-color imaging, confocal and STED. Maybe in the STED you cannot see very clearly. Every single spot in the confocal image is a two-spot image in the STED image. You can see here below some single origami structures imaged by STED in the two channels. And this is the average of these four, for example. So clearly we can resolve the two FRET spots. And then we can take a look if there is FRET going on. So we can do confocal FRET, of course. And you see, we clearly see anti-correlated signal jumps between donor and acceptor. These are kind of complicated anti-correlated jumps, because remember, we have two donors and one acceptor, but still you can see FRET going on. But sorry, and here you have two pairs of FRET at the same time, because they are inside the diffraction limited spot. And here you see STED FRET super resolved. Here we have the images, and now we basically what we do is we take several consecutive STED images, in this case 16 STED images, and then we can follow the intensity, for example, of this one, of this spot, and you see clearly anti-correlated jumps. Or in this case, we follow this one, and again you see anti-correlated jumps and so on. So this proved us that we can super resolve FRET events. Now we needed a method to apply this in real samples. And okay, these are simulations. Maybe I don't have time to show you the details, but the formalism of FRET that we use to compute FRET is this one developed by Tom Jovin, based on intensity and the so-called KT over KF method. It's basically a computation of intensities. You need to record three images: the image of acceptor emission and the donor excitation, the image of donor emission excitation, and the image of acceptor and the acceptor excitation. And all the other parameters are just corrections due to the setup that measure the crosstalk and so on, and the efficiency of excitation of each molecule. So here you see an example on microtubules. What we did here was just to label microtubules with the donor and acceptors all over it, just to have a subdiffraction scaffold sustaining FRET, and see if we can resolve that. And that's what you see. Here you see the microtubules super resolved by FRET, and you see by STED, sorry, and you can detect FRET on them. The other thing I want to show you here are these histograms. So it is important for this method that even if there is no FRET, just by fluctuations of intensity, you will compute some F value or some FRET efficiency value. And this becomes more critical when you do it on subdiffraction regions because you have fewer molecules. So you have just a few donors, just a few acceptors. So if you have one or two missing, the ratio of intensities will change, and then you will have some distribution that, let's call it a background distribution of F values. And this is the histograms that we call negative control here. We computed that on the microtubule sample, for example, doing samples labeled only with donor, samples labeled only with acceptor. Then we align artificially two microtubule segments, and we compute the F due just by random intensity fluctuations. That's that histogram. And then when you have real FRET, you have these other histograms, the ones on they call in blue. So it's important to measure both, and then you can set up a threshold. And of course, you will sacrifice some positive FRET sites, but the ones you detect in the end are clearly positive FRET and with a predefined statistics. Okay, so that's how it works. And finally, here we showed some examples on the MPS, the membrane periodic skeleton of neurons. This is a structure composed of adenosine, actin, and spectrin, and we can show that FRET takes place exactly over those rings, even though actin is distributed all over the cell. The FRET sites are located only, the FRET sites with adenosine are located only on the MPS structure, which is only resolvable with super resolution. Okay, with that I finish, and I'm happy to answer any questions that you may have. Thank you very much.
Thank you, Fernando, very much. Wonderful talk. There is a raised hand by Felipe Montesino. ¿Puedes habilitar a Felipe? Que el micrófono ahí. Está arriba de todo. Yeah. Sí. Okay. Felipe, I think you can now ask your question.
Hello. Hi, Fernando.
Hi.
Thank you for your excellent talk. I have a question about your microtubules that you showed in those cells, COS cells. I believe it's the TURF nanoscopy.
Yes.
So I wonder, how did you label those microtubules?
These were labeled with antibodies, and antibodies conjugated with DNA oligonucleotides.
Okay.
Or DNA paint. This was DNA paint experiment.
I see. The antibody is a large molecule.
That's
However,
right.
you're observing only the dimensions of the microtubule cylinder that you show are the actual dimensions of a microtubule seen, for example, with electron microscopy. So I wonder, why are you not observing a bigger structure?
We are observing a bigger. You see that histogram is the histogram of diameters that we measured. It's 41 nanometers. A clean microtubule has a diameter of 24 nanometers.
Yeah.
So
I see.
An immunolabeled microtubule is larger. It's about 40 nanometers. That's what we measured.
I see. Okay.
Yeah, it is larger. It is larger.
Thank you.
Nevertheless, the hollow core, you can also clearly see it because well, the antibodies evidently do not go inside the microtubules.
Yeah, I see that. Okay, thank you. That was my question.
Yeah, no problem.
There is another question here in the chat, Fernando, from Amit, and he says, what are the typical sizes of the fluorescent probes in nanoscopy? Good large sizes and steric hindrance of the proof affect the image evaluation of nanoscopy? Was kind of the same that Felipe asked.
Well, but I mean, that's a very important point, because nowadays the nanoscopy techniques are providing really molecular scale resolution. So we have microscopes and analysis tools that give us molecular scale resolution, but we don't always have probes to fully exploit those techniques, like just the question recently. We have a resolution of below 10 nanometers, but in this case we used antibodies that are 15. Okay, these were fab fragments, so these were a bit smaller, but still, the probe is comparable or worse than the resolution. So there is a lot of effort right now trying to develop small molecules that are fluorescent and specific. And there has been some success in some cases. But it's a very, laborious work. It's not like a universal platform like immunolabeling. Typically, you have to take drugs, like medicament drugs, that are specific to some protein and try to make them fluorescent. But normally when you do that, they are no longer specific. So it's really a tailor-made chemistry work. With DNA origami, with DNA, you have some advantage. If you have conjugation chemistry to place oligonucleotides, that's a bit more efficient, because then you have a very small oligonucleotide that can be a bit long, but still you put the complementary like this, and you put the fluorophore at this tip. So the fluorophore is really close to the structure you want to label, and the rest of the structure may be wobbling around. But you can see the improvement in resolution when you do that.
Okay, so Marilina is already there, so you can ask your question, Marilina.
Doc, I was wondering, I don't know if it is a silly question, but in the mean flux that you say that you illuminate and collect the photons, and then you change the positions and collect photons again. I was wondering, what about the photostability of the probe that you need for doing that?
Yeah. Okay, that's a good question. Actually, since mean flux requires much fewer photons than any other conventional nanoscopy method, it's suitable for more fluorophores. But because, okay, if you really want to reach the best result, of course, you need again the best fluorophores in order to get a few thousand photons. And then instead of having 10, 20 nanometers, you end up with 1 or 2 nanometer resolution. But let's say you have a probe that is not so good, and it only gives you 500 photos. Well, then you will get a resolution that is still way better than you would get with conventional nanoscopy, because you are more efficient using those photons. So
okay.
It's more flexible in terms of photostability mean flux.
Thank you, Fernando. There is more question, but we will skip for the at the end of the section for the breakout room, if you can be around, or I can place your information and the people can contact you.
Okay.
Thank you so much, Fernando. Wonderful talk.
Thank you. Bye-bye. Leonel, estás muteado. You're muted. We cannot hear you.
Sorry. Yeah, I was mute. I mute myself. Okay, so I think now you can hear me. Okay, so what I was saying is now I'm going to talk about a different talk, a different thing, which is basically related to spin microscope. The spin microscope here is regarding to the light shield microscope or single plane illumination microscope. And I will make a focus in particular on an instrument that we developed at the LFT with Nick Hede, the size beam. So basically, as Nick mentioned yesterday in his talk, the concept of the light sheet microscope is not new at all. Indeed, it was one of the first, or the first microscope to be able to do fluorescent. And the reason for why is because at that time, at the early 900s, it was really difficult to split excitation and emission. And the way that they found is what we know today as a light sheet microscope, they call ultramicroscope. And this technology was dormant for almost a century. And the reason for why is what Nick mentioned yesterday, because basically all the camera-based technology take a lot of time to develop. And basically until 1993, when this came again, or the people rediscovered this technology, and then by the work of Husk and many others, it got spanned to different fields, but in particular to the cell biology. So how does it work? Well, we can produce using different strategies, and I will address in a minute, a light sheet, and then we can image with a second objective, basically a full plane with a confocal resolution, or with a theta sectioning. And this has some advantages, and I will cover this. So we already know that basically when we use confocal microscope or wide field microscopy, we excite basically with a Gaussian beam areas which are outside of the region that we want to focus. And in the confocal microscope, what we do is basically we use a pinhole in order to reject the areas that we don't want to see, basically, and focus on our target. Well, with the light sheet, we can do this now because we have a sheet of light which is basically illuminating a full plane, and we can collect all the photons coming from this plane in a single shot due to camera. This has many advantages. The first of one is the speed, of course, because you can have the full chip of a camera imaging one single shot that can be on the range of millisecond time scale. But the other important thing is we are not illuminating nothing above or below what we want. And this is important for bleaching. So these are the two main advantages. There are different configurations of the light sheet. Basically, we can have double objectives with an orthogonal location. We can have single objective configuration. We can have mirrors, fibers, etc., that can produce this effect of the light sheet and the emission. But how we can produce the light sheet? Well, and Nick already mentioned that we can have cylindrical lens in which we can produce a Gaussian beam, but a flat Gaussian beam, and we can focus this flat Gaussian beam on front of the detection objective, and then we can collect the camera in the camera. The other strategy can be basically to use a scanner, so a mirror that can move a Gaussian beam across of our scanning optics, so the scanning lens and the tube lens, and at the end we will produce here on the other side, after it passed through the objective, a light sheet or a single plane illumination. So there are different advantages, disadvantages of these two. Of course, this is very cheap because you need only the optics. And in that case, you need the scanner. The scanner usually is costlier than the thing, but the important or the useful thing of the scanner is that, well, for calibration is very easy, because if you stop the scanner, you have a regular Gaussian beam, and this is very easy to basically make the focus with the second objective. The other important thing is that you are not illuminating areas which are outside of the field of view that you are imaging, because you can control how far you want to go with the scanner. And this is not controllable here, it's fixed. So when I joined the LFT, Nick was already, he already built this light sheet microscope. It was known as the upright SPIM. And in this case, we placed basically two objectives, one with a low numerical aperture and the other one with a very large numerical aperture, but low magnification. This is a 25x 1.05 numerical aperture, and the reason for why is because we wanted to do a single molecule experiment on this experiment in this microscope. Then we can split in the two paths and recombine them, and half of the chip of the camera will record one color and half of the other chip another color. Or we also did polarization, so it's very nice in this case to accommodate basically polarizers and do anisotropy measurement with this microscope. So here, I don't know if this is gonna work, but this is a small movie that you can see cells doing different things, moving. And you have different dynamics here. You can see, I don't know, paxillin forming here. You can have a cell dividing. You can have a cell migrating. So one of the big advantages of the light sheet is that we can cover all these different dimensions in times with a little changes in the microscope. So we can go to biomolecular dynamics, we can go to cell migration, we can go to cell division or anything, just because this is a very friendly setup that we can cover all these alternatives. Well, the upright spin was used in the LFD for a lot of application. Most of these related to single molecule experiment, and Nick did a lot of experiment regarding to the IMSD application, so he can measure the diffusion of a molecule. Indeed, he did it in the two channels, so he did it cross correlation, anisotropy measurement, which is a very nice work on the formation of the paxillin, and you can see there the homofret and how the homofret affect the anisotropy, which is very nice work. And then he did a really large field of view, and I will show how this can achieve. And also he set up an incubator in order to have very long movies of cells and different experiments. So if this is wonderful, why we want to stress or do something different? Well, as I said at the beginning, it has some constraints, or so difficult to accommodate basically this large detector detection objective, and we need to place our sample in some kind of a postal, because the light sheet cannot get on the bottom of a simple chamber. And for that reason, it was one of the main difficulties that we found in order to work in a regular configuration. But also we need to immerse our objective here. And this limits our possibilities in order to use, for example, toxins that can, I don't know, be hazardous for us, or they can distort or broke the optics that we have here. So at the end of the end, we stress our thinking in order to find a different configuration. And the main reason for why we need to change this, it was related to this effect, which is due to the way that we illuminate the cell. So we have a kind of a sagittal view, which restrict the possibilities to do correlation, such as the connectivity map that Enrico introduced the other day. And I hope if I have time, I can discuss a little bit. So one day, Nick come to my office and he said, well, I think I have an idea. The idea is the side spin. And what he thought is, okay, but if we can have a chamber in which we can have, instead of one window, we can have two windows, and we can put basically the light from the side and image from the bottom. And I said, well, this is a wonderful idea. But he said, well, we have a problem. And I will show in a minute what is this problem. The problem is we cannot get to the bottom because it will screw up our light sheet. Okay, so what is the solution? Well, we found a solution that I think is very interesting and is very friendly to work with cells and with organelles. And this is an interesting thing. So the last thing is, well, we need to design this lateral illumination, and these three things were part of a patent application that we placed a couple of years ago, and now is already set up in the US. And the setup was already covered by Nick, but I will go through very, very fast. We can do single photon excitation. In this configuration here, we were using a white light laser. So we need to basically have different filters. So we have a wheel filter here in order to select the different bands. And then we have some sort of pinhole and other optics in order to clean up the shape of the beam. Then our combination between the two photon and the single photon excitation, our scanner, the scanning lens, and then our objective here, and the emission objective here. So this is a very easy and very cheap configuration to do on an existing epifluorescent microscope as the one that we have here. So what are the problems of the refractivity index? Well, as I said before, if we approach close to the window, in the bottom window, the index of refraction will change from the up and the half up part of the Gaussian beam and the half down part of the Gaussian beam. And this will distort the formation of the light sheet. So for instance, what we need to do is basically put our light sheet, our sample up in the place where there is no problem with this. So what we found in order to do this was basically we can find some supports that can match the index of refraction of the media where we have our samples. And if we do that, this is solved. We basically did some calculation in order to see how much need to be this difference in the index of refraction between the media and the support in order to achieve a proper light sheet formation. And we found that this need to be on the order of 0.3. And trying to find it in the market, different resins that can be used. We identify a couple of them that match this requirement. And lucky for us, the collagen is a very useful biomatrix that we can use for this. And it's also can match our requirement for the index of refraction. So these resins are very simple to work. So here I place a box, and it's very difficult to see here, but this is a resin that we have to place in a different chamber. Of course, this chamber does not work. We can cure with a UV lamp. It's very simple, and we place this resin within a chamber with a 1 millimeter width. And then we jump to the point that, okay, we need a chamber with the two windows, and these were not available in the market. So I remember this day in the LFD, when Nick came to my office, we jumped to the little machine shop that we have there. We make it work the LED. We open a hole in a regular chamber. We glue a piece of glass, and we did the first test, and everything works. And this convinced us that this was the way to go. So then we produce a second generation by the help of Sasha Dvornikov with the machine shop, and you can see here the resin. And after a collaboration with Shivav, another postdoc at the Biomedical Engineering, he designed all these different chamber cutting with a laser machine, and it was very simple, easy, and fast to do it. So this is how we look like. We glue a glass in the bottom, and then we glue another two in each side, and then we have each of the chamber. We place some metals and some pins here in order to use magnet to support this pendant stage that is located in the XY stage. And on top of that, we put a piezo stage in order to move really, really fast our sample, because for doing 3D imaging here, we have to keep constant the distance between the excitation and the emission, and we move our sample. And considering that we can move with the piezo, we can go really, really fast. And I will show you this in a moment. So this is the performance of the light sheet in a solution. So we can reach a resolution of 0.7 micrometer using 0.3 numerical aperture in the excitation beam. And in the emission, we use, depending of the situation that we want to, or the problem that we want to address, but we can go for low numerical aperture to high numerical aperture with long working distance without any problem. So then we did the same calibration on the resin, and this is done basically here, and you can see that when we measure the PSF, we can reach more or less similar full width at Hala maximum that we have in the previous calibration. One problem that we have with the light sheet is that no matter what, we have this effect of the Gaussian beam, and we have basically distortion at the size of the focus, because of course here the Gaussian beam is broader. And this is how it looks like. Well, we have more or less these are beads. You can see that in the center they are very decent focus, but in the perimeter this is worse. So a way to solve this is a develop that Nick performed before, and this is the combination of an electrical lens with the rolling shutter of the CMOS camera. And I covered this at the beginning in the workshop. The first day, I explained that the CMOS camera can be run with a rolling shutter, which means that we can read the lines over and over from top to bottom. And if we coordinate this rolling shutter with an electrical lens that enable us to change the focus in the axial direction easily and fast, we can do what Nick showed here, which is basically we can move the focus of our Gaussian beam across the field of view, and at the same time take the best image that we can with our rolling shutter from the CMOS camera. So this is more or less what Nick found, and he can achieve more or less almost a 1 millimeter resolution improvement with the, of course, the reduction in the resolution in time, because we need to do many images across the full field of view. So I will go to the application. The first thing is, well, okay, but can we place cells on top of our resin? Well, the resin, unfortunately, are very hydrophobic, but if we activate these resins with the plasma cleaner, and then we put PMN and fibronectin, the cells can live and they can move without any problem. These are 3D stack done with the system. And these are cells placed in the inside of a matrix. And I said before that the collagen was a very nice support for our sample. And this is an ideal solution where you want to work in the 3D, because 3D is ideal because we are far from our bottom. So in that case, this helps us to avoid all the issues of the light sheet formation, and it's very good to analyze our samples. So these are, for example, the lamellar bodies labeled with the green fluorescent protein, and you can see the lamellar bodies moving, and these are mitochondria labeled with Tamari, and we did many more experiments with a different fluorophore. And this is an example that I like quite a lot, because we image basically the lysosome with lysotracker label for all the lysosome in a cell, considering that we can do really 80 planes with a 500 nanometer resolution in an intervals of 0.2 second. We can basically image all the lysosome within a small time, and this enables us to track all the lysosome in the cell. And this is what we did. This takes a lot of computational effort, because imagine that you have to track all the lysosome in these two cells, and you can see here all the trajectories. Then we calculate the frequency of the velocity for all the lysosome, and this was published in this paper a couple of years ago. But when we noticed that we can go really fast, we stress the system to be used in order to combine the technology with the flow chamber. So to use kind of microfluidic devices. And this is a very nice design, because we can go from the top and we can flow solution here. This is a very primitive, I will say, flow chamber. But anyway, it helped us to test. And these are beads passing through the volume, and we are scanning in 3D. And you can see that the up to more or less 3 microliter per minute to 4 microliter per minute, we can recover the 3D without any distortion. And when we go a little bit far, we can see that there is a sniff, which is basically due to the pass of the bead during the 3D stack. But this was important because a collaborator of Nick, Shivav, from the biomedical engineering department, was interested in the growth of bacteria in different support, and also in order to put to use this system to analyze different antibiotics and different molecules that can affect the biofilm formation. So we set up this on top of our microscope, and then he placed some bacteria on top of agar, and then he placed a flow. And what you can see here in the video is the formation of the biofilm over the time. And you can see single bacteria moving around and building up this biofilm over the time with a resolution of 1 second for the whole cytostack, which is very, very nice for dynamics studies. But then one of the important application of the light sheet was in the developmental biology. And in our configuration, the funny thing is, for most of the configuration, you need to basically do a major change or major tuning between the excitation and the emission, because we need to replace one and then match the other and everything. Considering that here we are working with a commercial microscope, the only thing that you need to do is to just turn around your objective for the new one with lower resolution, and then adjust the focus, and then do imaging and pass from the cell to embryos without major difficulties. So this is what we did. We have here zebrafish labeled with night red, which is a membrane dye that can go through the whole animal. And this is the light sheet passing through in the 3D stack. You may see basically different structure, like for example, the muscles, and then you have also the big vessels and veins and the erythrocyte passing through. And I will wait a second in order to pass the cytostack. You start to see these obscure areas here. These are due to the distortion of the image, because when the light passes through to the pigment that this zebrafish has in the skin, is distort completely the light sheet. And this can be avoided, or at least can be solved. Basically, if you, instead of having only one objective of excitation, you can place one from another, and this is already reported in many other examples in the literature. But considering that we can also go really, really fast in these large samples, I found very interesting that we can see, or if we can record the movement of single erythrocyte in 3D across the microcirculation. And this is what we did. You can see here these dark little things passing through here. These are erythrocytes. And we can track this erythrocyte and measure more or less the time that take to go from one place to another and do, I don't know, mean square displacement and all this calculation very, very easily. So finally, we basically decided to also check if we can do this high throughput imaging, and this is something that we are running now. The idea is that you can record one cell, move to another one cell in a chamber with certain condition, then we move to another and do the same, and move to another, do the same, and come back to the first one over and over and over very easily. And one of the good things here is we can reduce quite a lot the volume of our sample, which is very useful when you have to test drugs, which are sometimes expensive or difficult to produce. And you can accommodate here also microfluidic devices that you can replace the sample or change or make a gradient of concentration, etc. And this is something that we would like to keep working in order to development. So considering that I still have like the minute, and I want to stress why we finally designed this. And the reason for why we designed this microscope was to do spectroscopy, and I say before, to do connectivity maps in a classical view of the cell, which is in terms of the sagittal, basically the horizontal view of the cell. Enrico already introduced the concept of the path correlation, but what I would like to say here is that if we have stacks, and we take many of them, and we can do correlation between 2 points, and then we can do correlation of all the points around for all the pixels in the image. And at the end, if we calculate the path correlation, we may see that if we have isotropic diffusion, the image generated by this correlation that we call sprite is homogeneous in all directions. But if there is any kind of distortion for the path of the molecule in one direction or another, what we will have is an anisotropic diffusion, and this will be revealed by this sprite with a change in the shape of the path correlation function in two-dimensional. So to analyze this, what we use is the first and the second moment, and this enables us also to calculate the angle. And then due to these two values, we can calculate how distort is the path correlation function. And this enables us to calculate the eccentricity or either the anisotropy, which are two ways to measure this distortion, and also the angle of the movement. So the concept was already introduced, but is, well, we have some certain eccentricity. If we place a threshold, then we can place a stick, which give you the idea that there is a preferential diffusion in certain direction here. This is kind of a pipe or a tunnel that we draw here, and the molecule only can flow in this direction or if the molecules are bumping here, the average movement will be horizontal, and I will show the simulation in the next slide. So here we don't have labels for our structures. What we are trying to see is if a molecule which is not interacting with our structure can reveal the structure by the information that we obtain from the fluctuations. So this is kind of a method. So, and if we leave this going on, we then can do the path correlation, and you can see that we can capture all the things. This is just a cartoon to show the idea, but if we change the correlation, we cannot do correlation, for example, inside any of the pixels inside this box, because you cannot go more than 4 pixels far, and you will notice immediately that this is an object that is with an impenetrable barrier. Another interesting concept is the difference between the central moment, which is centered here in the path correlation function, and the center of mass of the path correlation function. And if we calculate this, we can see that there is a shift between these two values, and this shift in the center of mass is very useful in order to see the presence of the barrier. So we did some simulation, and this is a channel with 300 nanometer wide, and we fill up this virtually. Well, this is a simulation with a homogeneous concentration of fluorophores, and then we run the experiment, and then we do the calibration, we do the path correlation, and if we look at the path correlation function in different locations, you may see how it's deformated by the fact if there is a barrier or if there is an obstacle or anything. So here is the situation when you are close to the wall, and you may see that you have only half. So the preferential diffusion here will be along the axis. Here is in the center, so all directions are equal. And you can see here that if you move a little from the wall, you can immediately distort, and this is different to this one. So it's very sensitive to really little change. And the distance here is basically the resolution here is the pixel size. Then you can go inside the tube, the pipe, and you can see that the correlation is along as we expected, and in each of the sides is the same of the situation when you are in the wall. So we calculate the eccentricity, high eccentricity in the center and in each of the sizes. And if we place a threshold for the centricity, then we can do the connectivity maps. So to test our system, the first example that we thought is, okay, but we need to challenge our system. And a good way to challenge our system is if we have something that is not aligned, that can go around. And this is a GUV labeled with a dye. And if we are able to see the correlation due to the diffusion, the only way that you can see is every pixel should be tangential to the next one, because it's the only way that the dye can diffuse. And this is what happened. We see here the eccentricity, and then we see here the connectivity map without a major problem. So, but how data usually look like? And I like to show this because most of the people never estimate what we are seeing when we look at the original data. This is original data. I mean, this is what we are using for the correlation. And for me, it was amazing that basically we are looking at very dim information here. This is the background, but still we can obtain very valuable information, which is hidden if you look only at the intensity. These are then the connectivity, and I will show in the next slide using different part correlation how we can achieve different. Oh, yeah, sorry, I have here the movie that if you change the part connectivity with different part correlation distance, you can see different features in the cell due to the presence of obstacles and barriers. This is the same, and then I will stress at the end of this basically the value of the center of mass shift, and this is the center of mass shift, and the center of mass shift can show you immediately the occurrence of impenetrable barrier, and this is the cell. You can see the envelope, you can see the nucleolar, and then you can see this kind of trabeculated structure here, which we think this is the ER going around in the cells. It's funny because we can see things, but it's difficult to assign to something because we don't have a label for the structure. But anyway, I found very interesting this slide to share with you. So to finalize, basically what I want to say that the size beam solve most of the issues that we have when we start to do this experiment for lower volume, isolation of the optic to the sample. We don't have restriction to access from the top, which is difficult when the upright configuration. We can image our sample in the same plane or the same classical plane that we used to use in the confocal microscope. We can easily interchange different configuration for the sample as well as the objective, and it's absolutely compatible with any other super resolution techniques as you can imagine. So the final thing is, as a future perspective, some of these are already done, and Nick yesterday gave a beautiful talk about this multiple color imaging by spectral imaging using the sine and cosine, and this snapshot acquisition enabled us to image very large area with many fluorophore at the same time using the spectral phasor. Also, we did some experiment we never finished with the film, and they was asking this. Yes, it's very interesting, and it's more interesting to combine film with even spectral imaging at the same time. And we are very willing to try to test all this application for the high throughput. So thank you very much. I think we are very good on time. So I will stop my sharing, and I am open to answer questions if there is from the audience. Fernando, you have one down. Okay, well, there is some question here. So there is a question from Ramiro Quinta. Do you want to ask the question, Ramiro, by yourself? I think you are now allowed to open your microphone. Okay. So the other question, the other question here is, would you recommend spin for slower processes in order of minutes of hours, say a meet? Well, the good thing of spin is really the good performance in relation to bleaching, you know. So if you have to image something for really a long period, I strongly recommend. The other good thing is it's really cheap technology if you can build it, right? There are many commercial microscopes. They are not that cheap, but if you have to build it, you don't need really much optics. You need light. You need an excitation emission objective, you need a camera, and then you need really few optical paths in the excitation, honestly speaking, and in the emission too. Depending on how much you want to complexize the system can be more difficult. So the second question is, you say about you can do it. You can do it.
So it was about if, like, when you had showed the study of the biofilm thing using microfluidics. So I was wondering if, like, the viscosity of the medium, because biofilm has a very different viscosity, right? Does that influence your results? Because, like, viscosity is a function of, you know, the fluid flow pattern. So yeah.
You mean what you say is if for example, the change by viscosity can change the index of refraction, and at the end the
Exactly.
result. Well, in that case, we were using a buffer on top of the biofilm. So basically, we didn't change anything in the media. So the viscosity will be changed within the biofilm in a way, I guess. So I'm not an expert in biofilm, but so basically what we have in the solution and what we have behind was the match in the index of refraction. So we don't see any problem with it.
Okay. Okay. Thank you.
Oh, you're welcome. Any other question?
Hello.
Pablo. Miro.
Okay. Hi, Lionel. Excellent talk. I wonder if you could challenge your technique using a whole tissue, a transparent tissue, for example, with clearing technique or clarity that were developed by Frank Bradke.
We never tested, but I am pretty confident that we will be much better, because you saw in my 3D stack with the Nile red in an alive animal without any clearing, that you screwed up the light sheet when you pass through points we are really strong in scatter or in the absorption of the light, like the chromophore. So if you can clarify, all this will be out of your optical path, and this will help you quite a lot. But we never tested, honestly speaking.
And you think that, for example, in clarity, that is the technique that I use. I performed the transplantation using organic solvent, like tetrahydrofuran or chlorometano. However, in contrast, the clarity technique use SDS to perform the transplantation. So you think that your technique could use with this kind of old technique, clarity, and clearing that use organic solvents?
Yeah, I don't see any problem, because at the end, regardless of what you are using for the clarity processes, you place your sample in a different media, right? You place your in a buffer or whatever. So if you transfer this to the chamber, you can fill the chamber with whatever solution you want, and you will not be affected by the solvent that you use for the clarity or for any of the processes. I am guessing, because honestly speaking, I never work with any clarification. We always try to dive within the scatter and try to take information in the most natural situation. But this is the best that I can tell you.
Okay. My comment would be that if you use clarity, the sample will not be alive.
This is true. Yes, this is true.
Okay, so most of the techniques that you are using are for live samples.
Exactly. Yeah, I didn't say, but in the animal, you see the flow of the erythrocyte across the microcirculation. So these animals were alive. But also all the cells that I showed during the presentation were live. And it was one of the big objectives of this development to do live imaging. Right? So, Pablo, I think you have a question.
Yeah, I was thinking to reduce the background or in particular with the live cell imaging that you have, I was thinking if you can apply the process in the Luca was speaking in the morning with the film phaser to reduce the background according to how you collect.
In this approach, Luca was using lifetime measurement, and due to the step processes, he can split the photons we were bleached, or well, not bleached, but the lifetime reduced, from the photon where does not have any effect from the state, from the background. So you have the lifetime of all. So in here, everything that I show was just a single point intensity. So you cannot do that because you don't have this information. But I say that we can place a film camera, and indeed we did this experiment with film camera. The thing there is, if you have film, you will have the component of the autofluorescent. So you can consider this component in your experiment, and you can assign this background that you call as the autofluorescent. And for sure, you will have linear combination between background and your fluorophore. This for sure, because this will be related to the ratio between the amount of autofluorescent, which more or less will be constant along your sample, and the distribution of your fluorophore. That will be different for sure, because if it is a label for, I don't know, certain structure but not other, it will go to the nucleus or will go to the plasma membrane. But yes, lifetime will help you quite a lot.
Yeah, but
also spectra, right?
Yeah, but it wouldn't be so fast because you need to collect the photon or not.
But in our case, we will be even faster because Luca was doing raster scanning, right? Yeah. We collect all the pixels in a single shot, but we are not doing instead. We are not doing photo depletion, right?
Okay.
So actually, the camera for lifetime is faster than raster scan, much faster.
Yeah. You can obtain the full chip in the film within a second.
Yeah.
For the full chip, right? So
or even less than that.
Remember how was the size of the chip of the PCO camera, Enrico? Could you remember? It was a 20.
We have one size which is 512 by 512.
Yeah.
The PCO can reach 1000 by 1000 pixels.
1000
the exposure time is 10 milliseconds or something like that. So you have a lot of photons in 10 milliseconds, while in Luca's method, you have a microsecond or a few microseconds per point. So you need a lot of power.
Yeah.
I see. So in theory, it is possible to improve with the film. When you apply the film to your system, you would improve even more the quality of your image?
No, you will improve the number of things you can measure, the parameters.
Okay.
But the quality of the image is determined by the excitation and by the scattering. And so in that case, the clarity techniques can improve, but certainly you cannot follow the development of an embryo.
Yeah, exactly. Yeah, we're thinking about that also. If I wanted to long-term live cell imaging or some very fast processing with cells and culture, so that's what I was thinking about the background and to see the details.
Well, culture cells have no scattering, very little scattering. So yes, you can do for hours or whatever. But in an embryo development, you have a lot of scattering. So as it grows, it starts to scatter. But we have embryos measured for a day or several days. So that's a long period of time. And the damage is very, very small in the spin.
And the good thing is, considering that the size spin is built on top of a commercial microscope, it's very simple to have CO2, temperature control, everything, right? which help you quite a lot. In the other configuration, you need to really deal on where or how you are going to put all these things, you know, because it's hard. You have the constraint of the building of the microscope. Okay, thank you, Pablo. Fernando, I don't know if I saw your hand up.
Yes, I have a very short question. On your side light sheet setup, and you use this special chamber to compensate, to kill all the refractive index mismatches with the side windows. Could you do that by adaptive optics? So using a more conventional chamber and using light spatial modulator to pre-compensate the phase of the beam and obtain a good sheet?
It's a good question. I mean, in principle, yes. I mean, this is one of the advantages of the adaptive optics, because you can shape the different region of the Gaussian beam in order to get the best focus on the with the optics that you're using, right? Well, we never tested, but what I can tell about this is the work of Simon Lemans from the.
But this is done in scattering media. In the case of the stream, you know, where you put your waveform detector, it is more complicated than that. Okay, because the waveform waveform detector have to make the perturbation, and we do it through scattering material because we measure the fluorescence, but you can measure the fluorescence there too, but it will be more complicated. So adaptive optics is very nice if you can measure the wavefront, but or the intensity of the signal. It's clear to me how you correct point by point. You know, either you have the adaptive optics.
No, I thought of correcting. I thought of correcting just for the geometry of the chamber, and
okay. So that's sort of.
So as use any chamber, correct for the. Yeah, and that would be fixed. Like just for each chamber, you have some correction.
Yeah, I agree. It's a good point. Yeah, absolutely. Yeah. So if there is no more question, now we have a break. And basically the idea is that we will split with the speakers in single break rooms. So Luca told me that he will come back as soon as he can, because he was teaching in Italy. If he cannot make it now, maybe he can join with the person who wanted to discuss with him a little bit later. For those of you who want to join Fernando or me, we will start now the Zoom for 30 minutes, and we will recombine with all at 13:20 for the afternoon section. Thank you.
Una muchacha que quería ir.
Leonel, I don't know if you can listen to me, but you can pass me in the chat room where Luca is supposed to be. Maybe I can give some of the answers.
Great, Enrico. I will do now.
Okay, so I don't know if anybody will go there. Okay.
Pausa la grabación en YouTube. No, para así, pone pause ahí. Ahí va. Enrico, no hay nadie ahora anotado para ir a la breakout room de Luca, así que...
Bueno, pero la manera que dijiste, no va a venir nadie. ¿Qué dijiste que Luca no va a estar?
No, I know, but what we were saying is I will assign you to the Luca room